Hey,
Hello.
hi.
Hi, I'm Josemary. On behalf of my,
I'm Te-
what was your name?
I'm Tejas Borkar from IISER-Tirupati.
Hi, Dejas. It's nice to meet you. Thank you so much for
Yeah,
joining it.
yeah,
Great. So let's get started. So,
yeah. Sure.
good morning, everyone. I'm Josemary Cloud, and today we're interviewing iGEM team IISER-Tirupati. Did I say that correctly?
Yeah.
Okay, great. So would you like to tell us more about your iGEM team?
Uh, Sorry, can you repeat? It's breaking.
Can you tell us more about your iGEM team project?
Yeah. Actually, this year we have decided to work on the problem that is the AMR, antimicrobial resistance. As you know, it's like a upcoming problem which might come ahead in the near future. As due to the continuous usage of antibiotics, we are having a less stock of new antibiotics for the treatment nowadays. So we decided to focus on this year. So we would be working on reducing the efflux of these antibiotics in the environment through our engineered bacteria. So that's what it's a brief idea.
Great. Can you tell me a little bit more about the antibiotic degradation process?
Sorry.
Can you tell us more about the antibiotic degradation process?
Oh, sure. So actually, our project consisted of three modules. The first and the important one is the degradation module for the antibiotic. So in this module, what we will do is use the genes that are naturally available for the degradation of antibiotics. So currently, specifically, we will be working on the sulfonamides. So we will be using a two-pair gene system, which is SULX and SUL-R. That is a flavin-dependent oxidoreductase system. So it's pretty common system. So we will be using it to degrade the sulfonamides specifically present in the excreta, which we have been detected during our preliminary results and the outreach program during the initial days of our project.
Okay. And then, so you said you're uh, bioengineering your organism using the flavin oxidoreductase system. Are you bioengineering your organism in any other ways? Because I know you said there's three-step process degradation. First step. Can you talk more about the other step?
Sorry, it's breaking. Uh,
So you mentioned that you are bioengineering your organism with the flavin oxidoreductase system.
yeah, sure.
That's one of the components. So are there other ways that you're bioengineering the organism? Um, and
Yeah, actually, we are planning for its biosafety as well as the kill switch module. So currently, will be focusing on the avoiding the conjugation, what would be a major problem in this case, because if the genes, the resistance gene, get transferred to the other bacteria present in the excreta that we will be treating,
yeah.
so it would be of no use of doing the whole project. We are trying to avoid the resistance and then letting out the genes itself in the environment. So that's why we will be having a conjugation reduction module where we will code some genes, specifically the surface exclusion genes, that will avoid the conjugation, and thus our genes won't be transferred to other bacteria. And the third and the last module will be the kill switch and the death of the our engineered bacteria, so that after doing its work, it will degrade its resistant gene and will die off. This would be done by a user modulated kill switch.
Okay. That's awesome.
Yeah.
So you also speak about DNA degradation. This is part of the kill switch.
Yeah.
Okay, great. And then are you also programming for other bacterial cell death? Are you using other
No, we will be.
mechanisms for bacterial cell death?
No, actually, we will be using a user modulated PBAT promoter, which would be activated at different levels of glucose and arabinose concentration in the media, so that it's kind of regulated by ourselves So when exactly we want to kill our bacteria and after what time period of degradation or what percent of antibiotic degradation we want to kill, that's all would be user modulated by us. So
Awesome. I
yeah.
definitely want to commend your sustainable efforts. That's really wonderful that you know, you're encompassing the fact that it should be sustainable and keeping the environment healthy. How has your team been coping with your research during COVID?
Yeah, so currently we were completely focusing on modeling this stuff and getting like what aspect we can, what till what extent we can use this system, and through modeling we are trying to address this problem and check the success of this system, like all three modules.
Okay. Okay. And then along the way, what are some ways your team members have supported each other with methodology or with new modeling? That I know modeling might be different than being right in the lab. So, what are some ways you guys have helped each other through it?
So we have successfully modeled the conjugation system. So getting it known like what exact time period we have to degrade the antibiotic in the excreta, so that we know the exact window that we have before the first conjugant is formed. So that's all is done for as of for now, and we are like proceeding for rest modules and integrating everything.
Okay, great. And
Yeah.
then, if I could ask, for your team, what would you say has been the most enjoyable part of this journey?
So the most enjoyable part of this journey was our like initial days when we were able to do the lab work and preliminary work. Currently, we are enjoying it, doing everything online, holding up meetings and all. But still, the fun doing lab is at next level.
Good. I'm really happy to hear that. And is there anything you would want to convey to the audience about your project? Actually, we have someone who asked a question. You had any paper published on this? Has your team, or any paper that your team is referencing for your project?
We have referred almost like a lot of paper for this during our literature work study regarding this PBAT promoter as well as this Salex and Salah gene system. Actually, this Salex and Salah system is pretty new. This system we have adapted from a research paper from 2019, which was published. So yeah, we have referred a lot of research articles for now.
Okay. Does anybody have any questions they'd like to ask IISER-Tirupati? Do you have, how is molecular biology scope in the future? So do you have any commercial developments in mind to continue this in the future, especially because it's a three-step process? What does it look like?
The scalability of the project, right? So yeah, actually we have thought on the scalability of this project. So we are planning to have a treatment tank so that we can treat the waste, not only the excreta. As of for now, we will be working on the chicken excreta or the poultry excreta. But in the future, we are planning to work on treatment of the waste from hospitals because it's another rich source of antibiotic efflux in the environment. So we are planning for designing a tank which we would use to treat this antibiotics and get a safe manure from the poultry excreta.
Okay. And then at the treatment plan, how would you control for? I know you said there are multiple factors involved in the kill step. There's the actual the engineering, the bioengineering.
Yeah, so actually for as of for now, we are planning for a 3-stage tank where we secrete or synthesize the proteins which actually degrade the antibiotics, and then at the second stage we induce our kill switch, And after inducing the kill switch, the lysate which contain the degrading enzymes would be mixed with the excreta, and then after a certain incubation time period, so it would be free of antibiotics, and then it can be safely used as a manure.
Okay. Awesome.
Yeah.
That's really awesome that you guys have that idea of what a plant, an industrial plant, would look like to adapt this process.
Yeah.
Is there anything else that you would want the audience to know about your project and your team members?
Uh, so actually, we were like planning for the molecular cloud funding for sponsorship for a long time, and it's been a long time. We are trying to get the votes, but but we didn't get it. So it was a nice opportunity having a talk with you guys, spreading our idea regarding this antibiotic resistance. So if anyone wants to help us and fund us or sponsor us, so they can just contact us, or else they can just Google Impact Guru, which is our which is our keto funding. So there you can support the public funding, and they can support us there as of for now.
Well, you still have 3 opportunities to win our sponsorship.
Yeah.
So go out there and vote for
Yeah.
IISER team IISER-Tirupati.
Yeah.
All right, everyone. Does anybody have any last minute questions? All right. Well, thank you so much for meeting with me today.
Yeah, thank you. Thank you so much for giving us this opportunity to share our idea
No
with
problem.
everyone. Yeah.
No problem. It's our pleasure. So everyone go out there and vote for team IISER-Tirupati. Thank you for joining us.
Yeah. Yeah, thank you. Bye.
Bye-bye.
Have a good day.
You too. Bye-bye.
Yeah, bye.